opportunity to present this work. And I think this will be a nice bridge between the talks last night that focused on some of the broad themes, and then some of the later talks today when we talk about the specific therapies and treatment modalities, trying to bridge those with some concrete details in prostate cancer and what I think are some of the challenges of applying our knowledge to patients. I have the following relevant disclosures. So we're in an area where we have a massive amount of not just general genomic data, but prostate cancer specific omic data. The TCGA paper is in press at 300 samples, and I think probably the surest sign that we're near saturation is that there's actually very little novel information in the TCGA paper for prostate cancer compared to many of the other cancers where we're learning an incredible amount. And that's just in part due to both the large individual as well as some of the large collaborative efforts to profile prostate cancer. So I'm going to talk about a few of some of the near-term applications of prostate cancer genomics. I'm not going to focus on the top there about early detection and risk stratification at diagnosis, where both germline SNPs, urine-based transcript assays, as well as the prognostic assays based on gene or protein expression. I'm happy to talk about those after or offline. But I really want to focus on the last two, talking about basically the molecular subtypes of prostate cancer, and then how will these be applied and how are they being applied. So the prostate cancer has very robust molecular subtypes that'll be coming out in the TCGA paper, and I'll show that. And I'd say that, the good and the bad thing is we've known about these for several years now, but they're very robust and they're just as good as the molecular subtyping in breast cancer, melanoma, et cetera, that are commonly used every day, but these aren't right now driving patient management and specifically therapy, which is why they're not routinely used. The majority of prostate cancers, and this is from PSA Screen Caucasian series, different ethnic differences as well as detection methodologies, but about half of prostate cancers have a ETS gene fusion. The vast majority involve fusions of TMPRSS2 to ERG. TMPRSS2 is a strongly androgen-driven gene resulting in very high expression of these ETS fusion genes. They're almost nearly entirely the full-length genes, so you're overexpressing an oncogene specifically in prostate cancer. A variety of other alterations that are mutually exclusive and define specific subsets of the ETS negative group. SPINK1 overexpression, CHD1 deletion or mutation, as well as specific SPOT mutations, and then some small activation of the oncogenes. So I think we're not quite where we are in lung cancer, but I think we're getting close. There's also some things that aren't specific or mutually exclusive with any of their own alterations, but the typical tumor suppressors, P10, P53, and RB. This is the sort of the genomic landscape of localized prostate cancer from the TCGA. Just a few take-home points here, and I think one of the nice things about the TCGA is it provides a really nice reference data set with multi-omic, multi-platform data. So this is sort of just what I showed you in the other table, showing you these are cancers that have either an ERG, ETV1, ETV4, or FLI1 ETS gene fusion. These are nearly mutually exclusive with cancers that have an SPOT mutation, FOXA1 mutations, or IDH1 mutations. I'll come back to that IDH1 mutation, but that's a 1% type mutation. You can see that, interestingly, several of these other omic platforms, so this is methylation data, miRNA, et cetera, you can see that the methylation data is very distinct between ERG positive and ERG negative tumors, as is the miRNA data. So multi-omic data supporting these different molecular subtypes. One of the things that I think this sort of gets misconstrued in prostate cancer a lot is we think that prostate cancers don't have a lot of alterations, and that's in part because they don't have a lot of recurrent somatic mutations. The typical diagram when we look at cancers in these large sequencing papers is to show the recurrent somatic mutations, but you can see there's lots of copy number alterations. These are gains or losses here, loss in blue, gains in red. Several of these are more common in certain subsets of cancers, i.e. these losses here. But you can see that we have tumors that have lots of copy number alterations and those that are fairly quiet. Just getting to the IDH1, I think this provides a great example when we talk about rare alterations and what do we do with them. So this is actually from one of our papers that came out before the TCGA paper. We had a single case. This is using targeted next generation sequencing. This is a small targeted panel. This is a copy number plot. So gains would be up here, losses would be here. This is a relatively quiet sample. Has an IDH1. This is the R132H mutation that you see in brain tumors as well as leukemias. We look across all cancers that have ever been profiled. This is mutually exclusive, so we never see these in tumors that have either an ERG or an ETS gene fusion or an SPOT mutation, and it's about 1%. This is the IHC with the antibody that's available. It's used diagnostically in a brain tumor workup. This is a high-grade, this is a Gleason 9 tumor at prostatectomy, and you can see this is diffusely positive clonal, suggesting that this truly does define a unique molecular subset. TCGA data shows that these have high methylation. It's drivers. It's in young patients that don't have other alterations. So this is, I think, as bona fide as we're going to get is a true oncogenic driver in prostate cancer. The issue is it's 1%. So how do we do that trial? What do we do? There is an open phase 2 trial for this, for patients with this mutation in prostate cancer. It has not accrued very many prostate cancer patients because we're not sequencing these patients in the real world. So this is a concrete example of, I would say, an urgent sort of unmet need, and it's figuring out how do we get this testing done on patients to figure out if this is a subset that may benefit from therapy. And then just talking about how we're doing this and the different types of tissues and samples that are being assessed. And I think this is sort of a critical thing. We can talk, yes, genome sequencing, we can do a $1,000 genome, and that's true if you're doing germline depth on high quality tissues, or high quality blood DNA, not if you're doing formalin fixed tissue with 10% tumor content. That doesn't work at 20x or 50x coverage. So the different types of alterations from the different types of molecule that we're looking for, and the different technology that can be applied depending on whether you have high quality fresh frozen, archived FFPE tissue, or as we're increasingly seeing, and I think several of the talks today will focus on whether we can look at either plasma in terms of cell-free DNA, circulating tumor cells, and which techniques are appropriate, reliable, and can be performed in a clinical setting in a validated approach that can be used to drive therapy. And I think this is something that can't be overlooked when we talk about the technology that's available, particularly when we talk about how to get this to patients outside of specialized centers. And so this is sort of, if you want to take the extreme specialized center approach, this was a multi-institution study led by Rule Chennai and Charles Sawyer's. Paper came out in Cell this year of a multi-center effort to take real-time biopsies of patients with metastatic castration-resistant prostate cancer, in large part to get high-quality RNA for RNA sequencing. And patients underwent DNA sequencing of their exome on the germline side, tumor exome, capture transcriptome exome, and exome sequencing. So about 8 to 10 grand a patient is the actual cost of doing this, not what a hospital or a CLIA lab would charge. But it really gives us sort of a complete view of the metastatic castrate prostate cancer genome. And so many of the same alterations, and we've seen these from some of the other sort of single smaller studies, we see lots of alterations in the androgen receptor. This is something we don't see in the primary tumors, at least at a clonal or detectable frequency. P53, P10, we see the same distribution of ETS fusions. And I think there are some take-home things in here. One of these is that alterations are infrequent in CRPC in terms of recurrent alterations. So there is not a mutated tyrosine kinase at 10% that everybody's been missing. But again, I think there's some things that could be highlighted as particularly specific examples for where treatment may be important. So as was mentioned last night, the DNA damage repair pathway alterations, and particularly with the success of PARP inhibitors, there's now been at least 3 public presentations of complete durable responses to PARP inhibition in patients who have somatic BRCA2 alterations. So not just germline BRCA2 or BRCA1 alterations that predispose to prostate cancer, but somatic loss of function mutations in BRCA2 in particular. And so this is of the about 153 patients, about 23% have an alteration in the DNA pathway repair, DNA repair pathway. This is showing patients in red here. These are patients, so they have a variety of alterations. You can see BRCA2 is the predominant alteration, both 2-copy somatic copy loss. These are patients here with little double stars with a germline variant and then loss of the other allele. And then these are patients who have strictly somatic alterations in BRCA2. You can see the same thing with ATM, BRCA1, and a few of the other genes. So I think, and we're seeing this, and I think several people are, how do we target these patients? What's the best thing to do to look for these patients? Should you just look for patients that have a known germline BRCA2 alteration? You will get less patients than if you include those who have somatic BRCA2, but then you have to be testing tumor and not just the blood sample. Can you use the diagnostic sample, or does it have to be a real-time metastatic CRPC biopsy? That's unknown. Is it worthwhile to go after these other things that may be 1 or 2%? And then how can you actually prove if the drug works in this subset of patients, how can you actually prove that it works in these patients when you're going to have to get 30 patients in the treatment and 30 patients in the control arm? I don't know how we're going to get there until this becomes standard of care. You also have this group here that have the MSH2. So these are, again, somatic deletions in MSH2. So this looks just like the mismatch repair. These have mismatch repair number of mutations. This is the group that if you, based on the previous data in other tumors, this is the group that may benefit from checkpoint therapy inhibition right now. This is, again, 1%. It's reproducible across lots of people's studies. Emmanuel and I have been trying to find these patients, but again, this is not a group that's been routinely tested for. So how do we test for these patients? How do we get them onto the trials? Our group and others have applied this, looking at, I think the next step is going to be not just taking patients with CRPC, not just patients that are included in TCGA, most of which have intermediate to lower risk disease, but being able to apply this on diagnostic biopsies for patients who present with very aggressive disease, and using a more targeted approach when we know we can capture 95% of the alterations that are important. This is our study using a multiplex PCR-based platform, and not just to be profiled localized prostate cancer, CRPC that's been treated with chemotherapy, but we have some of the small cell and the more aggressive subsets that really haven't been profiled. One of the focuses of the West Coast Stream team, and I think it'll be nice to see, so we can sort of get a complete map of prostate cancer genomics, not just at sort of two extremes where we are right now. And just as a brief segue into that, and I know that several other people will be talking about this during the session, and so I think it's also really important to understand as we start to treat patients more that the biology that they started with may not be the biology that they end up, and that particularly that if we're doing testing, so single analyte testing, as we heard last night, just looking at the DNA may not give us the complete picture. So this is a small cell carcinoma, so small cell neuroendocrine carcinoma. The naming convention of these still very much in flux. It's a confusing issue for our clinicians every day. One of the things that we typically see, so this is that when we see this very aggressive small cell carcinoma, we see RB loss, P10 loss, and we always see P53 alterations. The group of 3, I would argue, means you have very bad, very aggressive disease, whether it is histologically small cell or not. This is an interesting patient. This is a patient who failed on ADT and then failed after abiraterone, underwent a lymph node dissection. This is the profile of that. This is the histology. This is not small cell. I don't know how I would classify this, but it's very aggressive. You can count the mitoses. This doesn't look like typical acinar adenocarcinoma, and it doesn't meet the histologic criteria of small cell. Nevertheless, this patient by IHC had very weak to patchy PSA staining, so very low expression of a typical AR target. Nevertheless, at the DNA level, you can see the AR amplification is actually off the chart here. Real high level of MYC amplification, a P53 mutation, and the T2ER gene fusion, which is strongly driven by androgen, is expressed at very low level. So high level AR, you may suggest this may mean AR is still important. We should continue to block this pathway. But the IHC as well as the expression suggests that this is a tumor that is having very little active androgen receptor signaling. So I think it's going to be particularly important in patients that have been treated to understand both the DNA and the expression. And then we're certainly raising the complexity of metastatic castrate-resistant prostate cancer. We now have biological evidence to prove what we already know, that if we treat with a single agent that is molecularly targeted, the tumor will develop resistance. We have proven that concept over and over again. Every time it's a Science or Nature paper, and we keep proving that point, we act surprised when patients recur. I would argue actually the best side that you have a good target is rapid resistance and mutation at the DNA level in something in that pathway. So these are patients, and I think when the study came out from Steve Bova's group, and it's been presented as sort of studying the natural history of prostate cancer. I think the important thing to remember is all these patients were heavily pretreated, multiple lines of anti-androgen and chemotherapy. So if you say at the very end of this, what does it look like? You will see lots. So this is showing alterations that are truncal. So they did a rapid autopsy study, took the primary sample that was left in. So the prostate was exposed to all of those alterations, and then multiple metastases, profile all of those and say, how are they related? And so a number of alterations are present in all the samples, but then there's lots of heterogeneity, particularly in terms of AR alterations. So patients that have failed multiple lines of AR therapy and chemotherapy have many clones, at least in their lymph nodes, and I think people will talk about this, also in their circulation as well. So the idea that a single agent will cure this patient is likely is not going to happen, although I'd argue that we know that already, that patients who have numerous metastases with numerous circulating clones who failed all of our standard of care are going to be the most challenging to treat. And we're seeing this is some work we've been doing on CTCs, where you can see the same heterogeneity of CTCs, not just the traditional cytokeratin positive, CD45 negative circulating tumor cells, but you can detect clusters, you can detect cells of the same size as a white blood cell, those that lack cytokeratin expression but express the antigen receptor. And so we're seeing this heterogeneity, and I think it's unclear sort of what's the best approach, the best molecule, and even the best understanding of how we want to predict what treatment response we're looking for. And so again, people will talk about this later, but just looking at circulating cell-free DNA, this is a way to capture the DNA, and it should be a representation of all of the clones that are in circulation, or at least those that are apoptosing or dying and releasing DNA. This is work by Gerda Tar, just showing this is something you can do this serially, and I think Emmanuel will show some of this data as well, showing that you can have lots of different heterogeneity in the clones that are detected, and these may vary based on treatment. And so again, we don't have a good idea of what is the best thing. Should we be doing metastatic biopsies? Should we look at CTCs? Should we look at cell-free DNA? And I think probably before we can decide that, we have to decide what is the question? What do we want to be predicting? What is a good biomarker to be looking for? And whether that's just strictly a biomarker of treatment response, or whether that informs on the mechanism of disease. Oops. So I think the genomic medicine, I think we have to understand the basic molecular subtypes. These are things that can be determined very easily up front at diagnosis. And in our experience, these do not change during progression. We have rare oncogenic targetable lesions in prostate cancer, if you want to call them the IDH1, RAF, et cetera. But I think we're going to have a paradigm shift in drug development to hit the common drivers, loss of tumor suppressors, etch gene fusions, et cetera. We don't have, as Oliver said, we don't have predictive markers for prostate cancer last night. We haven't looked for predictive markers. We actually, at Michigan, we've actually started the first biomarker inclusion criteria trial is ongoing, and I think it's the first in prostate cancer. So we've done one. So it's not too surprising that we don't have a lot of predictive markers. And again, I don't think we know what the best tissue sample is. That cell-free DNA, circulating tumor cells. What's the best one to do? And particularly in terms of doing that and then converting that into an assay that can be done across all centers. And I think the heterogeneity says that we're going to have to use some kind of combination therapy, and all this has to be done with the understanding of making sure that patients get the best treatment for them. So let me just end by thanking if I can get this to work, the members of my lab and some of the funders that have supported that work. Thank you.
Use this one?
Okay. Okay. Thank you very
that
much.
Works. That works.
Okay. Thank you very much. Very nice speak, and nice to see you again in this meeting. And so my question is since Dr. Cooper published a paper talking about the heterogeneity of prostate cancer, even for the same patient on the same section, since prostate cancer have multiple loci. So I have a question about the heterogeneity. So in terms of it, so could you please comment on the diagnosis by markers and the treatment? Because it's so heterogeneous, and it makes our diagnosis and treatment so difficult. So could you comment on what can we do to to efficiently diagnose prostate cancer, and how to target the treatment, maybe in a personal way, or personalize the treatment, something like that.
Yeah, certainly the multifocality at diagnosis, as sort of somebody who looks at this more from the molecular side than the anatomic side. When I started doing anatomic pathology, I'm kind of, we presume as pathologists that we know which is the focus that's going to drive biologic behavior. If there's two foci with the same grade, we say it's the larger one, that's the one that we pick and we do our molecular testing on. You have no way of knowing if that focus has grown slowly over 20 years and the other one is rapidly growing. So we don't, I think, have a good handle on that. We actually have a grant to do this, to go back to look at when we have the patient's CRPC sample, go back to their diagnostic sample and map out all the different foci and see if we can figure out the common patterns of spread. The work by the ICGC, I think that the challenge of that is they sort of pick these really big tumors to assess heterogeneity, and they found lots of heterogeneity. knowing that prostate cancer can be multifocal, we know that we could have two tumors that grow right next to each other. You know, that the previous work where people have picked small tumors spatially distant proved that those are genetically distinct. So, and again, I don't know if we can go right now off of the diagnostic sample to say this is the relevant genotype that this patient is going to have at the time of relapse. I think in some patients it's easier when you know that they have, you know, Gleason 9 disease at diagnosis, very high grade disease. It's fairly obvious what the index focus is. I think that can inform, but I don't think we have a conclusive answer to say, yes, we can use this diagnostic sample to predict how the patient is going to respond, particularly once they've been treated with anti-androgen therapy.
Or how can you give us any insight on how we can differentiate lethal versus non-lethal prostate cancer? I mean, these genomics that you're talking about are for the patients with advanced hormone, you know, castrate-resistant prostate cancer, but so many patients are undergoing surgery now who may not even benefit from it and just have morbidity with them.
Yeah, so I mean, you know, there are numerous expression or protein-based prognostic indications. I think that the challenge is right now the majority of them are being used to reassure patients that they should go on active surveillance. By clinical characteristics alone, their risk of having what the diagnostic companies have defined as bad pathology is less than, say, 5% for the vast majority of patients. We're not seeing urologists use this in patients with 6 cores of Gleason 3+4. That's where you should be using these tests, if they work that well, to say, yes, you need therapy or no, they don't. Where they're being used is patients who have 1 core, Gleason 6, less than 5%, and the test says that no, they don't need therapy, which is what we would have predicted without the test. There's certainly some of the work from the prostatectomy-based tests on trying to identify lethal disease. I just think that's a harder space to be in because it's harder to say you have Gleason 9 disease, T3A, but this test says you don't need any therapy. I think that's a harder sell to people to get them to do. So I don't think we have, and none of them are predictive yet. There's not good an, you know, the use of prognostic markers in cancer is almost zero. Predictive markers to predict response to therapy is much higher. That's why Oncotype breast has such great uptake versus Oncotype prostate. So I think that has to be solved. It has to be done in clinical trials and proven that these predict response to therapy. And I think that's when people will use them as a prognostic test, even though they're more predictive.
Along that line, why is KI67? Because you alluded to transition, all these other things are correlated with growth.
Yep.
Well, the best marker is KI67, and I don't see pathologists. It seems like that ought to be the first commandment. To measure KI67.
I said that at a Bar Harbor meeting a long time ago, and Jonathan Epstein got it right behind me and said that's the one marker he wouldn't pick. So
that's because John is a pathologist.
So I mean that
he deals with dead tissue.
So
who deal with the life is the issue. A cell has to proliferate in order to kill you.
So I can, you know, so the challenge with using KI67 diagnostically is it's just challenging to use that reproducibly in a clinical environment. The breast cancer group has this all the time. It's just challenging to use, but that's why Myriad's test, which grinds up the tissue and looks at 30 markers of proliferation at an RNA level, is a pretty good prognostic test.
But if something has every molecular change known to Jesus,
yep,
and it has no KI67 ability to grow, it's not going to kill you.
Yep.
And something that has no molecular changes that has a growth fraction
I completely agree. So I can just say, you know, so that's what Myriad's test. So part of the reason why it doesn't get used is because if you do a test of a RT-PCR based test and you do KI67 and you put them into the same multivariate model, the RT-PCR based test comes out as a better predictor. So that's the justification for that being used. I would argue if you look at them, they both perform pretty similarly. And the KI67 test costs about $50 versus a $4,000 diagnostic test. But nobody's making money off the KI67 test. So that's why it's not used. And it's challenging to use because most prostate cancer also has the proliferation rate is so low that it's 8% versus 6% versus 5%. Yeah.
I think you agree. One quick last question.
Yep.
Gosh, the ERG story started a lot of this work. You were there.
Yep.
And yet the ERG story still seems confusing to me. What is ERG doing? Why is it involved 50% of the prostate cancers? And yet we don't really, or at least I don't seem to understand exactly what that molecule is doing in the patient.
So the functional study, I mean, this is, part of the reason why we're not good at developing drugs in prostate cancer, because we don't have good functional models of, one of the 12 commonly used prostate cancer cells has an ERG fusion, despite the fact that 50% of tumors in every cohort does. So it's challenging. When the clearest example is when you take VCAP cells, which have endogenously have the fusion, and you take it out, these cells differentiate. They express PSA. They also express some of the neuroendocrine markers when you really knock it out. So I think it's basically just keeping the cell in a de-differentiated state. It is an early lesion. It doesn't drive, so many people have tried to look at this as a prognostic marker. That wasn't when we were trying to find it, that wasn't what it does. It doesn't drive prognosis. It's just an early molecular event. And I think, it's similar to, again, if it was a tyrosine kinase, we'd be having a very different story, but it is a transcription factor that is involved at the earliest stages of, and I've been criticized for saying whether it's truly driving cancer, whether it's driving invasive cancer. I think it drives the invasive process. So making malignant cells because of their ability to invade, not their ability to proliferate. And so I think people are now developing some better mouse models, and some with the organoid work, I think we might have a better idea of what the function is. But
Great. All right. Well, thank you very much. Terrific. I'm going to